Many experiments on the International Space Station are intended to learn what we can about the conditions of long-term spaceflight as a part of the preparations for future missions out into the solar system. That includes experiments that use the human crew members as test subjects and those that focus on the millions of other living things on the station, the microbes, which share the station environment with the human crew. Commander Terry Virts will be taking samples for the experiment Microbial Observatory tomorrow, and today we're joined by the principal investigator, Dr. Kasthuri Venkateswaran of the California Institute of Technology and the Jet Propulsion Laboratory in Pasadena, California, to learn more about it. Hi, Dr. Venkateswaran. Thank you for joining us today.
Hi, how are you? Thank you for having us on the air.
Thank you. We really appreciate you joining us to talk some about this experiment that they're going to be working on. I know today Terry is actually reviewing some of the procedures for that, and he'll begin doing some of the samples tomorrow. So let's first just talk about, where do the microbes on the space station, where are they?
They are everywhere. Like, we breathe and then we inhale and exhale microbes every day, So International Space Station is not a different environment either, other than microgravity. So they are everywhere. We have to look for it.
And where do they come from?
Yeah, that's a great question. So normally people say that it comes, when you are living in your house, the microbes comes from outside. But in the case of International Space Station, you don't have a football field, so the dirt comes in and all. But actually, the human are the walking fermenters. So they hold, say, 10 to the 14 cells, microbial cells. When compared to their own eukaryotic cells, they are about 1,000 times more. And it might come from the Earth through the cargo and the human who are traveling between them. So the main source are human, as well as the stuff comes from the Earth as a cargo.
Okay, thank you. So if we can't control the microbes that are present, why is it important that we identify and monitor the microbes?
It is not that we cannot control. The thing is, it is different from what we are seeing in the Earth compared to what is in a closed environment. Because NASA has taken utmost care to have appropriate air environmental system of a closed system like in space stations, to see what can be there in the air of the space station, which is a very closed system where a lot of things are being inhaled and exhaled by the human present in there. So it is not that you cannot control, but you need to know what is in there, so that for a long period of human habitation in a closed system, you can learn from there and avoid a kind of prevention, so that if you monitor what is in there, you can prevent easily. And we should not forget the microbes are integral part of the human, so there are a lot of beneficial microbes out there that helps us also. So we are not living in a sterile environment. Okay, we are not transgenic human. What that means is we are not made up of one single thing. We are made up of multiple things. So human knows how to live with things together, a kind of immune system they have to develop, so that for some other unwanted thing they can fight. The microbes out there can fight from other bad microbes also. So basically, maintenance is a key part to live and have a better living conditions in a closed system.
Okay, great. Thank you so much for that information. So I know Terry's going to be gathering samples tomorrow. Can you describe for me the on-orbit part of this this experiment? How and where are they going to be gathering, or where will he be gathering the samples?
I think you might have shown some of the places where we are collecting samples. So we have awarded this experiment through NASA Space Biology Program to use the state-of-the-art molecular techniques to see how the microbes are, there in a set of locations. For example, astronauts are using the exercise platform, so they sweat a lot. So that means they also, shed a lot of microbes. So that kind of place is where a lot of activities going on are the main target to have, and also day to day they have to eat, so there are certain specified area where they can eat and then chat. So that is the other place where people can shed stuff. So a dining table, and this particular experiment is on the US side only, so we are having the nodes 1 and 2 and 3 are covered of the US side, as well as some of the exercise place. And we also have other experiments which are on the way to collaborate with the European and Japanese people to measure the microbial diversity of their section of the space station also.
Okay, so now we know where, and I think you've also described why you chose these locations. The samples are being returned on Dragon. Will you be analyzing these, or how do you analyze the samples?
Yeah, so for the past 10-15 years, NASA is monitoring based on the traditional culture-based analysis. So we all know that you can get only 1 to 10% of information if you're using the traditional microbiological methods. So the state of the art has evolved from traditional microbiology using the genomic analysis. So we are using the gene-based system to get the genetic fingerprinting of these things. So that would help us to see whether any particular cargo is taking a particular kind of microbes all the time. So you have to repeat again and again the same sort of methods so that you understand where to control and mitigate the plan. So the state-of-the-art molecular technologies are very powerful and not time-consuming, and also not laborious, and also analyze a lot more samples. So we are using molecular techniques.
Great. And how do you anticipate on using the results of this study, on orbit and also on Earth?
Correct. So there are a couple of things. Number one is, if you know what are the things out there, whether that is beneficial to humankind or the one that is not good and harmful, and sometimes under microgravity some changes might happen. If such kind of mutation is happening, then you will know what needs to be targeted and eradicated using appropriate cleaning regimes, as well as some sort of mitigation plans can happen. So this is the first step towards achieving that particular goal, how to maintain a better living condition for the astronauts for a longer period of time. So right now we are getting data for 2 to 3 years, and that can be used to suggest NASA how to build appropriate environmental control system, thereby containing certain microbial parameters. And also, as I said all the time, there are certain many beneficial microbes out there that helps us to produce it for tomorrow's food processing. For example, if you are going to have a human presence in faraway planets, one of the biggest thing is how do you keep the food and replenish it. So some of the beneficial microbes that you see there might be useful to the conditions that helps them to process such kind of things and then get benefited.
Well, thank you very much for joining me again, Dr. Venkateswaran. It's very interesting research, and we look forward to learning more about the findings that you have throughout this. Thank you very much.
You're very welcome, and a special thanks to the Commander Wirth, and hope to get more good samples tomorrow.